All right, why don't we go ahead and get started today. Hope you guys all had a nice long weekend. Today we're going to be following up on our discussion from the last time when we were talking about T cells. And in particular, we're going to talk a little bit about T cell memory. And actually, before we get to that, we'll spend a little bit of time talking about B cells and the response against influenza. So you guys have all heard about vaccines. You go out and get your flu. How many people here have actually gotten a flu vaccine this year? Ah, good. So hopefully there's herd immunity. Nobody will get sick with flu here because a lot of us have been vaccinated for this. And we'll talk a little bit about the concept of how that plays out. We'll also talk about gamma delta cells. So you guys remember when we were talking about T cell development in the thymus, there's that subset of T cells that come out if rearrangements on the gamma and delta locus happen before you get productive rearrangements at the alpha and beta loci. And so the result of that is that you get these gamma delta cells that come out. And I didn't spend a lot of time talking about the function of those. We'll spend some time today talking about those cells. And then we'll focus on these natural killer cells, or NK cells, and talk about how they're involved in essentially an innate-like immune response. And then at the end, we'll talk about the fact that T cells, you know, we've heard about them being able to recognize peptide antigen in the context of MHC, but they can also recognize lipid antigen that's presented by these specialized types of class 1 MHC molecules called non-classical MHC. All right, so the last time we were talking, one of the concepts that I brought up towards the end of that lecture was this concept that memory B cells, but not naive B cells, are capable of participating in a memory response. And the reason for that is that if you have a primary response, let's say that you have let's say this is a bacteria, or this could be a virion, and that virion has associated with it a variety of different antigens that are on the surface. So this would be considered a polyvalent set of antigens because there's multiple antigens that can be recognized by B cells. So here's a naive B cell. It comes along and it has a B cell receptor that's rearranged that allows it to recognize these antigens on the surface of this virion. And so the consequence of that is that you get B cell receptors are clustered. That results in a positive signal being propagated inside of that B cell and then allows that B cell to undergo clonal expansion and to differentiate into a plasma cell, and you make antibodies. But the kinds of antibodies that you make in this response from a naive B cell are relatively low affinity. You guys have heard this before. And then on top of that, they don't undergo any sort of isotype class switching. And so the result is that you generate a lot of low affinity antibodies, mostly of the IgM isotype. So those are not particularly effective at getting rid of this bug, but at least it gives the immune system a chance to try to catch up with that infection. And so we also talked about the fact that once you've had this particular antigen in the system, over time that response is refined. You have somatic hypermutation that takes place, and you have isotype class switching, resulting in, for example, IgG, as well as higher affinity antibodies that then have a much stronger response to that particular virion. Okay, so what happens to these naive cells is kind of interesting. So here you have these antibodies that have already been made in response to, say, the first time that this particular bug has been encountered in the system. And the result of that, as I mentioned, is that you don't activate these naive cells. So these naive B cells have on their surface this Fc gamma receptor 2b1, basically binds to the Fc tail of these pre-existing antibodies. And if that happens, that sends in a negative signal into this naive B cell that says, even though you're capable of recognizing this particular microbe with your B cell receptor, we don't want to make a response. And so what's the logic behind this? Why would you want to set that up? Well, it makes more sense if you've already got pre-existing antibodies. It doesn't make a lot of sense to generate a naive B cell response because these guys have already gone through the whole process of maturation in germinal centers. You've got the appropriate isotype that's being produced, and you have high affinity antibodies. So it doesn't make sense to try to redo that. So this Fc receptor, if it's recognizing these pre-existing antibodies that can bind to this microbe, then that sends that negative signal, and the cells they won't respond, these naive cells. So that allows you to have a memory response. Now, in contrast, memory cells also have these Fc receptors on their surface, but when they bind to these memory antibodies, these antibodies that have been produced previously, that does not send a signal that turns off any activation. So in this case, the memory cells are re-engaged, they continue to become activated. Some of those will differentiate into plasma cells, and you'll have a memory response. And in fact, those memory B cells can exist inside of our immune system for essentially our life. So we know some examples of people that have been immunized or say they've gotten exposed to certain types of pathogens 50, 60 years ago. They still have memory B cells that can recognize that particular pathogen all those years later. Now, why is this important? Well, obviously, as I said, the rationale, if there is such a thing, for making this sort of response and for setting up this negative signal in these B cells is that it doesn't make a lot of sense to make a new naive B cell response when you already have pre-existing antibodies that are perfectly functional. But this causes a problem, and this is in particular with influenza. So some types of antigenic challenge or microbial challenge, an example of this is measles virus. They don't have a whole lot of change in their antigens, so they don't have this kind of antigenic drift that's associated with their ability to infect. But on the other hand, influenza, so the flu bug does. And so the reason why you guys have to go and get this flu vaccine every year is because there's antigenic drift and antigenic shift. These changes, and we'll talk about more about that on Friday when we talk about virus responses. But the point of this is that, let's say that you have these 4 different glycoproteins on the surface of a virion where you've made antibodies against those. So let's say this is the first infection, and you have 4 different antibodies that are generated against these 4 different antigens. So the consequence is that you can produce a productive response during this primary infection, and then you can deal with the bug and get rid of it. You'll get sick for a few days, but for the most part, you'll be able to clear that out. Now, the second time you get challenged, let's say that it's not exactly the same virion, the same virus that you encountered the first time. Let's say that it has undergone some sort of change in the glycoproteins that are present on the surface of the virion. So you have 3 of them in common with the initial infection, but now one of them, through mutation or what have you, has changed, and so the consequence of that is now you have these protective antibodies against these 3 existing antigens, but you don't have an antibody against this new red antigen. So what's going on there? Why are you not getting a response to that? So first of all, these guys are protective, so you're probably not going to get particularly sick because this is a memory response. But what's the problem? Well, over time, as you start encountering new variants of influenza, you see that the antigens are changing. So what's actually presented to the immune system is changing. You would think, well, now it makes sense to set up a naive response because we haven't seen these antigens before. But because of what I was just telling you with respect to these naive B cells, if there are pre-existing antigens or antibodies against the existing antigens here, they're going to shut off naive B cell responses because of the fact that this virion is polyvalent. It has a number of these different antigens on the surface, and the result of that through this Fc receptor binding is that that's going to shut off any sort of naive B cell response, and so you're not going to generate antibodies against these novel antigens that are coming up as a result of these changes that are taking place over time in these influenza viruses. So with time, you get to a point where you're losing more and more of the original antigens to the point where there are none of them that exist anymore. And at this point, your immune system can now mount a response because there's no pre-existing antibodies that can bind to the virion and shut off a response of these naive B cells. And so now you start to generate a new set of protective antibodies. But of course, over this time, because of the fact that there are a lot of these antigens that are shared between different strains of influenza, your protective antibodies actually start to go down over time. So even though you have a memory response, because of the fact that you're shutting off a naive response against these new antigens, you can't effectively mount humoral response. And so this is called the original antigenic sin. This is a concept that was brought up in the early 60s that says that, as a consequence of subsequent exposure to these virions that are changing over time, you actually start to lose protective immunity to that. So any questions about this? Yes?
In regards to the previous slide, if you have passive immunity transfer via something like breast milk, does that also shut down naive B cell response?
So would passive transfer of antibodies from the mother's bloodstream through the placenta to the fetus, and then of course into the child, would that shut down any naive B cell responses in the infant when they're challenged? Yes, it could, but of course the antibodies that are coming across should be protective because there's no, so whatever antibodies that the mother has, they should basically put the kid, somewhere in here, unless it's a completely novel strain, which is unlikely. Any other questions? Okay. So what about the memory T cell? So we've talked a little bit about the response of the B cells, and I mentioned the last time that you also generate this specialized population of T cells called memory cells. And I wanted to show kind of what the different phases of an immune response might look like in response to, let's say, viral infection. In this case, infection with cytomegalovirus or CMV. So if you look at the kinetics, what you see is that the virus, let's say that you're infected here at day 0, and you can see there's a really rapid response. There's a rapid increase in the level of infection. But then over time, you start to generate a T cell response. So this is the number of T cells that goes up that are specific for that particular virus. In this case, these are CD8 T cells. And they can recognize a particular epitope on this particular strain of CMV. So what you see is that there's this rapid increase. This is the process of clonal expansion that you guys have heard about. And this gets to the point where eventually the T cells are capable of controlling the response. So the pathogen then starts to go away. The infection is now controlled by specialized T cells we talked about the last time. What do we call them? The effector T cells, right? So they go from a state of, in this case here, they would be naive cells at the very beginning of this response, and then they can turn into these effector cells, which in this case are primarily going to be cytotoxic T cells. That allows them to control the infection, and then the virus goes away, and the immune system kind of returns to homeostasis. And the way that it does that is it undergoes something called clonal contraction. So that population of antigen-specific T cells, they're specific for CMV, that population needs to go away. And we call that clonal contraction. And if this process doesn't happen, what do you think would happen? If you don't have the ability, let's say that once you clear a virus out, or a bacteria, what have you, if your immune system is messed up in some way and you can't, your cells don't undergo clonal contraction, what do you think would happen over time? Yeah?
You wouldn't have a secondary response?
You might have a problem in terms of a secondary response, but on top of that, if you don't do this, you're not going to maintain any sort of homeostasis. And in fact, there are diseases, one is called ALPS, we'll talk about later in the course, where there's a problem in this process of clonal contraction, and so eventually, because you're constantly being exposed to different microbes, eventually you start to generate these huge lymph nodes and a big giant spleen, and usually this is lethal. So kids that get this usually don't make it past their 10th birthday because it's just such a dramatic effect on their immune homeostasis. So you need to have this contraction, otherwise you'd have a bunch of cells that you really don't need because you've already been able to get rid of that infection. Now the important point, and I mentioned this the last time, is that the number of precursors here at the very beginning is maybe, let's say 100 in your body. At the peak of this response, this may be 10 to the 6, maybe even 10 to the 8 cells, all of which are specific for this CMV infection. And then it comes back down to a set point of maybe about 10,000 cells. And those cells, because of the fact that there's an enhanced precursor population, there's more cells that are antigen reactive, then if you get infected by that same virus down the road, you have a bunch of these cells that are memory cells and can quickly convert into these effector cells to be able to deal with the challenge. So this set point is comprised of what we call memory cells, and these memory cells have certain features that are different from naive cells. So for the most part, they're expanded, so there's a greater precursor frequency, but they also have the ability to respond more rapidly. They proliferate more rapidly, and they generate into effector cells expressing the appropriate cytokines and having the appropriate effector response much more rapidly than you have with naive cells. And if you actually look at these guys, some of the things that you can see on the surface, and we immunologists like to use antibodies and facts to be able to define these subpopulations. But an example of this are on the naive populations, you can look at certain surface markers. So CCR7 is one of these chemokine receptors that we've talked about that's involved in getting these T cells into lymphoid tissues. And then L-selectin is involved in keeping those cells in these lymphoid tissues. CXCR3 and CCR5 are other chemokine receptors that are more involved in getting the cells out into the periphery, so where they can do their job. And so what you see is that on the naive cells, you have expression of CCR7 and L-selectin on the surface, but when they become effector cells, they no longer need to be inside the lymph node. They need to make their way out to be able to bring their effector function to bear against that particular infection. And then as they convert into memory cells, they have this sort of mixed phenotype. So they don't really look like naive cells. They don't look like effector cells. So they start to have different surface marker expression, and they're a unique population that is distinct from either the naive population or the effector population. Now, in addition to that, there's actually a couple of different forms of these memory cells. So if we look at their production, here you might have, say, a naive T cell that is stimulated by a dendritic cell. The result of this is that, of course, it undergoes clonal expansion. And some of those cells are then going to turn into effector cells. In this case, they might be cytotoxic T cells that can go then kill off that virus. But it's thought that at least some of these cells, these naive cells, can directly convert into these memory cells. And those memory cells then, once the antigen goes away, then they can be reactivated to be able to respond. But they actually differentiate into two different subsets. One are called the central memory population, and these are expressing on their surface CCR7, which is, as I mentioned, involved in getting these cells into the lymph nodes, and it retains them there. And then a second population are these what are called effector memory cells, and these effector cells don't express CCR7, so they're not retained inside of these lymph nodes. Instead, they're essentially tissue resident, so they're present at the site of the infection. So you might find these, for example, if you had a flu bug that the T cell was responding to, you might find these in the lungs interacting with the epithelia inside the lungs. And the reason for having these two different populations is that upon subsequent re-challenge, so if you're infected a second time by that particular microbe that induced the response in the first place, you want to, number one, have essentially a pre-existing population of effector cells that can then quickly deal with the infection. Okay, they may not be long-lived. They usually don't hang around for very long, but they have kind of an early response that can control the infection until such time as you have these central memory cells that can convert into effector cells. And so this is a population that will undergo a lot of clonal expansion, be able to deal with that secondary challenge, and then be able to eliminate the infection. All right, so then, as I mentioned, some of these memory cells are being produced from naive cells directly. And so the result of that is you get these two different populations of memory cells. Some of them are also the result of having undergone, they've differentiated into this effector cell population, and then they can differentiate from that point. Once the antigen goes away, they can then differentiate into these memory cells themselves. So they've previously been an effector cell, and then if they don't die, so a lot of the cells are going to end up dying off during that process of clonal contraction, but if they don't die, they can then become these quiescent memory cells. Okay, any questions about memory T cells? Okay. Now, one of the key points about these memory cells that we know is that memory is not dependent on antigen, in the sense that it's not dependent on that antigen staying there. Now, why is that important? Well, if you think about it, this is a population of cells that has previously seen antigen, but there's got to be something inside the body that says, you're a memory cell, we need to maintain this population in case we ever have some re-exposure to that particular microbial infection. And so there's obviously signals that need to be there to tell the cells, where they have to go, whether they should survive or die. And we know that for memory cells, they do not need to get this continuous exposure to antigen. So once they've been activated initially from the naive state, converted into a memory population, then they don't need that reactivation through the antigen receptor in order to drive or to maintain their survival. Instead, they depend on cytokines, and in particular, they depend on interleukin-7 and interleukin-15. So if you knock these out in mice or you block them using antibodies, the result is that you lose your memory populations. So you need to have those cytokines around to maintain those populations of memory cells. So how do we know this is true? How do we know that this memory population is independent of antigen stimulation? So you need antigen stimulation early on to activate the naive cells, but how do we know that you don't need it to maintain those cells after the response? And I should say this is an important point in the early 90s, because there were two different groups that had opposite results. One group said that, no, you needed antigen exposure. In fact, this guy went on to win the Nobel Prize for the discovery of MHC restriction. So his lab was, coming out with some data that supported the idea that you needed to have antigen stimulation to maintain memory. And then a paper came out from this fellow, Rafi Ahmed, in Nature, basically showing that, no, the work from this other guy was all bogus. We have the right result. Okay, so basically what they did to show this is if you take a mouse and you infect this mouse with LCMV. So LCMV is a mouse virus that's often used for experiments. The result is that these mice will develop lifelong immunity against LCMV. And we know that because you can take memory cells. So these memory CTLs, and again, some of the markers that we were talking about, that would be one way that you could show that. And then you could take the T cells out of these guys and then show that there's maintenance of these memory populations. But again, you wouldn't know necessarily whether or not there was antigen stimulation that was required. And the reason why I say that is because you've infected the mouse has been able to reject the virus infection, but you never know for sure that there might not be some reservoir of LCMV somewhere in this mouse. It could have gone latent or something like that. So they did a couple of additional experiments. One was basically to take this mouse, infect it, and in this case, basically remove the T cells from the spleen of this mouse and put it into a new mouse. So this new mouse has never been infected with this virus. And then 1 to 2 years later, you can take the spleen out of this mouse, and then you can find that there are memory CTLs that are specific for the LCMV. So you know that there's a population of cells that has become memory cells, and even 1 to 2 years later, at the end of the life of this mouse, it still has this population of memory cells that are specific for LCMV. All right, and then a third experiment, basically the same sort of what we call an adoptive transfer experiment. So you take T cells from a mouse that had been challenged with the virus, put that into a mouse that's naive, it's never seen the virus, and then challenge it. So in this case here, we're just looking at in vitro experiments to look to see whether there are memory cells there. But here, you actually challenge this mouse, and in this case, you use a virulent strain of the virus, which, if it was not protected, if it didn't have memory cells, the mouse is going to die because of the virulence of this LCMV strain. So the result of that is that if the mouse has been challenged with this virulent strain and it's gotten these memory CTLs, even 1 year later, it still provides protection to this mouse. So even though this mouse has never previously had a memory response, this acute challenge does not kill the mouse. On the other hand, if you fail to give the mouse memory cells, so here, this is a naive mouse that's never been challenged with LCMV and it's never gotten these memory cells, the result is that mouse will then die off because of the virulence, and it doesn't have a pre-existing memory population that can then protect it. All right, so that shows that you do not need antigen in these mice here in order to maintain that population of memory cells. That's entirely being driven by these cytokines. So as I mentioned, one of the features of differentiation of T cells in the thymus is that you primarily get these alpha beta T cells that go through the process of positive and negative selection. But there are also some cells that come through that we've talked about called gamma delta cells. And I mentioned that the gamma delta cells are a bit different in several respects. Number one, they don't undergo positive and negative selection. They drop off. So once you get the rearrangement in the gamma and delta chains, that then diverts that T cell from from the thymus, and then it goes out into the periphery. In addition, I mentioned that they have limited TCR diversity, and that's because of the fact that there are just not as many junctions. There are not as many gene segments that can produce different TCRs in response to recombination or rearrangement. And then on top of that, their specificity. So alpha beta T cells are primarily specific for peptide antigens, but in this case, these gamma delta cells are specific for these phosphoantigens, which obviously they're distinct from what the alpha beta cells are recognizing. And in this case, these cells are sort of innate-like in the sense that they're not recognizing antigens per se, but these phosphoantigens, which are essentially intermediates of biosynthesis inside of certain microbial pathogens. So our bodies don't produce these kinds of phosphoantigens. Those are produced by microbes. In a sense, what these T cells are recognizing are this unique population or this unique subset of antigens that are not peptide antigens. So it's very innate-like. Now, in addition, these gamma delta cells, because they're not expressing this chemokine receptor, CCR7, they don't percolate through the lymphatic system. So they make their way throughout the body and tend to show up in places where there's an inflammatory response going on. And the reason for that is that instead of expressing CCR7, they express chemokine receptors that can drive them to these sites of inflammation, like CCR5. And so the consequence then is that if you have some inflammatory response going on, these cells are going to divert to that particular area in response to that chemokine signal that gets them there. Once they get there, they kind of work sort of like standard T cells in the sense that they can express cytokines like interferon gamma and TNF, and then can promote, say, a Th2 response or a Th1 response or a Th1-like response. And they can also secrete certain proteins like granulysin, which is kind of similar to when we were talking about perforin. The result is that it kills the cells. So they have sort of a CTL-like, helper-like activity once they get to the site of infection. So they play a pretty important role in host defense. Obviously, if they're recognizing some of these innate signals, these phosphopeptides or phosphoantigens, they can then be directed to control those kinds of infections. But another example of this is that the gamma delta cells can essentially aid in a host response by recognizing a specialized subset of what we call non-classical MHC class I molecules that are called these MYC proteins. So an example of that is shown here, where you have, say, an epithelial cell infected with a bacteria, and the bacteria is taken up inside of the cell, and it's present in some of these endosomes. That then is a stress to the cell. So as a consequence of this stress, the cell starts to upregulate these non-classical class I MHC molecules that we call MYC, and that then sends a signal that allows this gamma delta T cell, through its T cell receptor, to recognize this MYC protein. And the consequence then is that the gamma delta cell then kills the epithelial cell. And one thing that's kind of an interesting side point about this is that not only do they kill this epithelial cell, but they also provide cytokines and the appropriate environment that seems to promote the production of new cells. So you don't just kill the epithelial layer, as a result of this kind of stress, but instead you actually help to promote regrowth of that layer. So in a sense, the immune system is not simply inducing the death of these, or trying to get rid of these bad things, but it's also trying to promote repair, which actually turns out to be a pretty important point we'll discuss more when we talk about autoimmune disease. Okay, any questions about the gamma delta cells at this point? Okay. So now I want to talk about these natural killer cells, which, again, sort of like these gamma delta cells, they're quite a bit, they're similar to T cells in many respects. They have the ability to kill, so they can use similar killing mechanisms like you have in CTLs, but they're, again, not recognizing peptide antigens. So they are much more innate-like in the sense that they can recognize if things are bad, if there's stress that's taking place inside of the cell that might be infected. And I want to show a quick movie on this.
Of all the immune system soldiers, natural killer cells are the most aggressive. They are lymphocytes with no immunological memory and are part of your innate immune system. Natural killer cells make up about 5 to 15% of the total circulating lymphocyte population. Natural killer cells are called natural killers because they do not need to recognize a specific antigen before releasing their toxins and destroying a viral infected cell or cancer cell. They target tumor cells and protect against a wide variety of infectious microbes. In several immunodeficiency diseases, including AIDS, natural killer cell function is abnormal, thus allowing the viruses to multiply inside of human cells. Natural killer cells kill on contact. The killer cell binds to its target, aims its weapons, and then delivers a lethal burst of toxins that produces holes in the target cell's membrane and creates events that destroy the cancer cell membrane and cleaves DNA into many pieces in a programmed cell death, that is, apoptosis manner. After releasing its lethal package of chemicals, the natural killer cell can detach from the cancer cell and kill many other circulating abnormal cells. Indeed, research has shown that most of the tumor cells that enter the circulation are destroyed during the first 24 hours, and only a few cells succeed in establishing themselves in distant organs from the primary tumor, which is called metastasis. Natural killer cells may also contribute to immunoregulation by secreting high levels of influential lymphokines. Therefore, research to develop natural products to stimulate the natural killer cell activity, especially in immune-suppressed patients undergoing chemotherapy for cancer, or in any viral disease such as AIDS or bird flu, where immune suppression occurs, is very important in the regulation of aspects of the immune system which affects our overall health. Finally, we come back to the beginning of our knowledge of the natural killer cell. Here lies a natural killer cell in a lawn of sheep red blood cells. These cells are perfect targets for the natural killer cell, possessing no MHC1 receptor. The wrap of natural killer cell is fully vented on the surrounding sheep red blood cells. The cytotoxins completely destroy the sheep red blood cells, leaving clear plaques around the natural killer cell. There is no apoptosis event, just direct cytotoxicity. The natural killer cell is one of the key soldiers in the fight against viral and cancer growth.
Okay, so let's talk about natural killer cells. Okay. That's bad, okay. Anyway, so basically what the movie shows is that these natural killer cells are really important for recognition of viruses and bacteria, and they're also really important for controlling tumors and can prevent these cells from going outside of where those cells would normally exist and result in metastasis. So these cells, as I mentioned, they're very similar to T cells in the sense that they have cytotoxicity, they can kill if they recognize something bad in the cell, in the target cell. But they don't have T cell receptors. They don't go through the process of positive negative selection. They may start out in the thymus, but they actually exit pretty early on in the differentiation. And what makes them different is they do have receptors on their surface, but those receptors are not antigen specific. Instead, they have, for example, the ability to bind to certain innate molecules. They have the ability to bind to MHC, and I'll talk a little bit about how that works. But each of these NK cells has a unique repertoire on its surface of these NK receptors. And the important point is that, first of all, we know that there's greater than 30 different kinds of NK receptors, and a particular NK cell doesn't express just one type. It has multiple receptors on its surface that can then control the activity of that NK cell. And as I'll show you in a minute, those receptors can both be inhibitory, so they can block the activity of these NK cells. They can also be activatory and promote their activity. So an example of this is, here you have a cell, an NK cell, and on its surface it has an inhibitory receptor. This inhibitory receptor we call NKG2D, which in this case here sorry, this is, I'll talk about this in a minute. This is CD94 NKG2A. Here's the NKG2D. But in this context here, there's an interaction with the healthy cell, and that interaction between this inhibitory receptor and MHC on that healthy cell basically shuts down the activity of this NK cell. So why does it want to do that? Why does it want to bind to MHC if it's not a T cell receptor and it's not looking for peptides? Well, it turns out that in tumor cells and oftentimes in certain virus infection, a target of that particular pathogenic situation is to try to reduce the level of class 1 on the surface. So as you guys remember, we have MHC class 2 and class 1. MHC class 2 is expressed where? Sorry? Yeah. On professional antigen presenting cells. But what about MHC class 1? Everywhere, right? So all of our cells have MHC class 1. That's important because it allows our CTLs to kill if there's some viral peptides or something. But these viruses are really clever, as are tumor cells. And what they do is they try to outwit the immune system. They don't want to get killed by CTLs. And so to do so, they target the expression of these class 1 molecules. And so they're thinking, well, if we just we can hide out inside of the cell by just downregulating the expression of MHC on the surface. So that's where these NK cells come in. So if they, for example, find out that there's no expression of MHC on the surface, the result is that they will then kill those cells. All right. So, and then another thing that they have, so that's through this inhibitory receptor. So basically, if you have class 1 there, it shuts down this NK cell. But then, as with these gamma delta cells, one of these NK receptors, this case NKG2D, can bind to MYC. So I was telling you before that MYC are these non-classical MHC molecules that are upregulated in response to stress. So if a cell is infected with a virus or it's becoming oncogenically transformed, the result of that stress inside of the cell leads to this upregulation of MYC on the surface, and NKG2D then sends a positive signal into this NK cell. So you have a positive signal promoting NK activity and a negative signal that's lost because MHC is downregulated, both of which allow this NK cell to then kill those cells, and that gives these NK cells a way of getting rid of this virus. Yeah?
Is there a special reason why the MHC class 1 is downregulated when the cell is infected?
Is there a reason why the infection can lead to MHC downregulation? Yeah, so actually there's a lot, if you look at viruses, viruses are really good at mutating and coming up with new forms and picking up host genes and so forth. And they're loaded with all sorts of different host response genes where they want to try to interact, they want to basically manipulate the immune response. So they'll take advantage of certain transcription factors, for example, convert them in a way that they repress MHC expression. So that's actually part of their evolution. And there's essentially been a huge amount of coevolution between our immune response, all the genes that are involved in our immune response, and then these different pathogens that we have to deal with, to the point where our immune system, for the most part, is a good thing, and it helps us maintain survival amongst this sea of bad stuff. But at the same time, it can then lead to autoimmune disease and autoinflammation. So how does this work? How does this negative signal actually come about? So one of these inhibitory receptors is called NKG2A, and it's paired on the surface of an NK cell in the context of another molecule called CD94. So normally, cells are going to have HLA on the surface, and the result of that, if you have class 2 on the surface, is that's going to send a negative signal in this NK cell to prevent it from killing that cell. So what is it recognizing? So it's not actually recognizing directly, standard class 1, which would be, in humans, HLA A, B, and C, but instead it recognizes a non-polymorphic class 1 molecule called HLA-E. So how does that work? So how does it know that there's downregulation of some of these guys, like HLA-A, B, and C? The way that happens is that here inside of the ER, you have, obviously, MHC, in order to get to the surface of the cell, it has to get into the secretory pathway. So it is co-translationally translocated into the ER, and in order to do that, it has to have this hydrophobic leader sequence. You guys remember back to Bio 99, where they talked about this in gory detail. So to get expressed here, it has to get across this membrane, and it uses that leader sequence. And then, of course, once it makes its way into the lumen of the ER, that leader peptide is then cleaved off. So there's a leader peptidase that gets rid of that. And this class 1 molecule called HLA-E then can pick up that little leader sequence. So it doesn't bind to anything else. It doesn't bind to other peptides like HLA-A, B, and C, the standard class 1 molecules. But instead, it can only bind to this particular subset of peptides, these leader sequences from standard class 1, A, B, and C. So once it does that, then it can then traffic. If it doesn't bind to peptide, just like any other MHC molecule, it gets degraded. But if it does bind to this peptide, this leader peptide, it will make its way through the trafficking system, the secretory system, out to the surface of the cell and be presented there in the context of this leader sequence peptide. And so if it's there, then that sends a negative signal through the CD94 NKG2A inhibitory receptor to the NK cell. And so the NK cell says, okay, fine, this is a normal cell. It's expressing HLA on its surface, and everything is fine. So, but the important point of this is that, you know, some of these viruses have tried to get a little witty, a little smart. And instead of just sort of giving rise to this bulk downregulation of class 1 on the surface of the cell, they try to target specific molecules in an effort to basically say, well, you know, HLA-A such and such is really good at presenting my peptide, so I'm going to shut that one down specifically. And so they try to upregulate some molecule that's going to shut down one particular MHC allele. And so as a consequence, our immune system has co-evolved what are called these killer cell immunoglobulin-like receptors, or KIRs. And basically what the KIR does is it's kind of like a T cell receptor. It doesn't undergo the process of, it doesn't look like a T cell receptor, but what it does is kind of similar, and that's that it can bind to the surface of class 1 MHC and then recognize what MHC molecule is there. And again, just like with the other NK receptors, there's a whole group of these guys that are expressed on NK cells. And so in this case, this is showing, you know, the hot dog in a bun model that we've talked about before, where you've got the peptide strung between these two alpha helices. And here, this is the footprint. Basically, what a standard T cell receptor would bind to is in this square, and it's sampling both these alpha helices as well as this peptide. But these KIRs really don't care so much about the peptide that's here, and they really just want to bind to this alpha helix, and in particular on this position 80, because this can detect, individual KIRs can detect specific MHC class molecules, and if they're downregulated, these are all inhibitory receptors, so if that particular molecule of MHC is not present on the surface of the cell, that sends a signal into the cell that says, okay, I'm not getting this inhibitory signal anymore, let's kill the cell because it's downregulated one of these specific HLA molecules. And in particular, this tends to be more specific for the HLA-C types. So these guys tend to be the ones that are favored. And reasons for that are not entirely clear. It could just be the process of coevolution with the kinds of viruses that try to attack this class 1 expression on the surface. All right. So let's hear more about these.
The natural killers per cell surface that regulate the recognition phase consist of two types. One is composed of the receptors that are homologous to the C-type lectins and is called the natural killer receptor complex. The other is composed of immunoglobulin-like domains called killer cell immunoglobulin-like receptors, also known as KIRs. These are shown on the natural killer cell surface as the forms KIR3D and KIR2D. However, the predominant control of the natural killer cell activation occurs through the C-type lectin receptor, CD94-NKG2 heterodimer. One complication in understanding the natural killer cell activity is that the same structural families, that is the CD94-NKG2 natural killer receptors, contain both activating and inhibitory receptors. The NKG2 proteins differ for the inhibitory structures of the CD94-NKG2 complex.
You got that?
Signaling by the inhibitory natural killer cell receptors suppresses the killing activity of the natural killer cells. The receptor for the target cell is the major histocompatibility complex 1, also known as MHC1. Natural killer cells will not kill healthy, genetically identical cells with normal expression of MHC1 as seen in other normal cells of the body. Therefore, the binding of the inhibitory natural killer cell receptor to normal MHC1 sends a negative signal to inhibit signals from the activation natural killer cell receptors, which are bound to the target cell activating ligand. However, if the MHC1 is 1, not dense enough on the target cell, or 2, if the target cell has processed antigen and displayed it in the pocket of the MHC1 illustrated here by the shaded red-yellow ball, or 3, if the conformation of the MHC1 proteins have been sufficiently altered, noted here by the stripped MHC1 proteins, then the binding of the altered MHC1 to the inhibitory natural killer cell receptor will send a positive to the activation natural killer cell receptor complexed to the target cell activating ligand. It is this positive signal that initiates the remarkable cytotoxic machinery of the natural killer cells.
It's so dramatic. I just love the see, I gotta have a little soundtrack going behind me or something to keep you guys awake. All right, so any questions about natural killer cells before we move on? All right. So the last thing I wanted to talk about is lipid recognition by T cells. So again, you know, we've focused really on the recognition of these peptide antigens, but there are some alpha beta TCR expressing T cells that can recognize certain lipids on the surface, and in particular, a set of lipids called glycolipids. So normally, and this is a result of another set of these non-classical MHC molecules, class 1 molecules called the CD1 molecules. So CD1A through C. Basically, what would happen is that normally you would be, you know, in a non-infected cell, you have endogenous glycolipids that will bind to these non-classical CD1 molecules. And then they traffic, so they go through the standard secretory pathway, make their way into the ER through the Golgi, and then to the surface of the cell where they can then interact with T cells. But in this case, because these are self-derived glycolipids, you're not going to have T cells that are going to recognize that. And why is that? What would happen to those T cells? Yep. Exactly. So those guys actually, you know, we normally think of them as being removed by peptides, but in fact, those endogenously produced peptides can also serve in negative selection. So if you have T cells that are, you know, overtly reactive against autoglycoantigens, then those are going to be removed. They're going to die by negative selection. So what happens? How does this protect us? Well, if you have, let's say, a certain kind of microbe, in this case, these glycolipids are produced by these mycobacteria, sort of like we were talking about with leprosy. And they make their way into endosomes, lead to, you know, this intracellular residence inside of these cells. The glycolipids that are produced by these bacteria, those can be recognized by certain T cells, and they then make their way into the class 2 pathway. So basically, here's a vesicle that's got class 2 present in it, where you normally have processing of class 2. But you also have these CD1 molecules, and they can then basically kick off the endogenous glycolipid, replace that with one that's produced by these mycobacteria, and then shunt that to the surface of the cell where it can then be recognized by an alpha beta T cell receptor. And so the result of that is that this alpha beta T cell can be recognizing these mycobacteria, the fact that the cell is infected with something, even though it's not recognizing peptide antigens. In this case, it's recognizing glycolipids. And so the consequence of that, of course, is that you can, you know, for example, kill the cell. So if this is a CD8 cell, it can act as a cytotoxic cell to kill these guys. This can also lead to the generation of effector cells, the whole process of clonal expansion and elimination of these mycobacteria. So any questions about that? Okay, so I'll see you guys on Friday, and we'll talk in detail about viral immunity.